This is the first of a two-part set of talks, initially looking at entomological field techniques, and then followed up by a second part looking at the use of Pantheon analysis software. So the first thing I want to do is to introduce myself. I currently work for Natural England. I joined the Natural England Field Unit as an entomologist in 2016, and I've currently moved to a different post within Natural England, working in citizen science. I've been involved in entomology in one way or another for around 25 years, and fortunately still am through work and outside of work. I first got involved in entomology many years ago, carrying out butterfly transects as a volunteer, and then I managed to eventually become a freelance entomologist, doing work for a number of government agencies, conservation charities, and consultancies around the UK. I'm incredibly proud to say that I learnt my entomological trade volunteering at the Liverpool World Museum's entomological department. I used to visit every Thursday as a volunteer for about 10 years, and I met some of the people who are on this call tonight, such as Brian Formston there, and many others, and got a really good grounding in entomology. In 2006, I joined the Field Studies Council, and I ran 3 funded projects there, including one that was a 4-year project on entomology called the Invertebrate Challenge, which worked with a whole range of people to learn the skills needed to become entomologists, studying a range of taxa. During that time, I authored a couple of county atlases for Shropshire on crane flies, which was the second one I'd done on crane flies, and one on shield bugs. And we also were able to commission work on Acheliot Hymenoptera and Microlepidoptera of the county too. In 2020, I published 24 new species of cranefly to Science from Cameroon, following a Winston Churchill Fellowship I'd done in previous couple of years. And I've also gone on to identify new species to Science from Corsica. Outside of Natural England, I'm a coordinator and national recorder for the Cranefly Recording Scheme, and I also teach biological recording of invertebrates for MMU and the Field Studies Council. Okay, so what are we going to talk about tonight? We're going to talk about the rationale of structured field entomology. We'll look at survey techniques and habitats, and we'll look at how you would carry out a survey that would produce enough data to inform Pantheon software of invertebrate assemblages and potentially other entomological outputs. Now, if you've never heard about Pantheon, don't worry, we'll go into that in a little while. For those of you who are going to be joining the second webinar in a few days' time, there will be a little bit of homework for you to get familiar with Pantheon, which we'll come on to a little bit later. We're going to look at ethics and regulations in regard to collecting specimens and why we do it, why we need to do it. We'll look at the best techniques to use in the field, and we'll assess what level of information we need to be able to get a useful analysis out of Pantheon. So there we've got the date of the second webinar. If we get through this one and you still haven't signed up to it and you think it might be for you, please go ahead, and we'll see how we go this evening. Any questions, as Leanna said, fire away, or keep them for the end, and we'll hopefully come back to them. Okay, so Pantheon. Pantheon is an analytical tool which has been developed by Natural England and the UK Centre for Ecology and Hydrology to assist invertebrate nature conservation in England. It basically identifies invertebrate assemblages that give us some idea of the favourability of triple SIs originally, and so it was designed to help monitor those sites, but it's since gained a much wider range of uses as more information has been coded into it. And as I say, I don't propose to go into any depth about Pantheon tonight. That's something we'll have a deep dive into in the next webinar. But you need to be aware that it exists, and it's the main bit of software that people use to assess sites for terrestrial invertebrates. Right. I think probably the best place to begin really is to have a very brief think about the ethics of invertebrate collecting and why we do it. When I ran my project at the Field Studies Council, this is a conversation I had with a number of people. There are a lot of invertebrates that you can identify from photographs, but the vast majority of them you have to capture and kill. Let's not beat about the bush, that's what we have to do to be able to study them and identify them and to find out stuff about them. So voucher specimens form the basis of a vast scientific archive. Those of you who have been into Liverpool Museum and or any other similar establishment will have seen the vast array of specimens there, and these are absolutely vital for scientific study. Most taxonomy is based upon the ability to study these specimens. That is changing a little bit, and we'll come on to that later on in the talk, but very much the basis of modern taxonomy is access to those specimens in museums and other people's collections. Certainly DNA studies are altering perceptions on how invertebrates evolved and their relationships, but as of yet, we're not at a stage where DNA will answer all the questions that we pose in terms of taxonomy. Most species fundamentally can't be identified in the field or even by photographs. I get lots and lots of photographs into my Twitter account for the Cranefly recording scheme, and I think with 20 years experience I can probably identify about 30% of the UK's craneflies by very good photographs, which means obviously there's a vast majority there that you need to collect and look at under a microscope to be able to identify. And also invertebrate sampling, it has a really, really small effect on population numbers compared to other forms of consumption. So what are those other forms of consumption? Well, some of them include the birds and other animals in the UK, and also other invertebrates, which eat an awful lot of insects, flying insects particularly. So roughly 99% of a swallow's diet is flying insects. They eat millions of flies, mosquitoes, agricultural pests, and it's estimated that there are around 1.5 million swallows, and if each ate just 350 insects per day, that would mean more than 500 million insects consumed just in one day. So absolutely mind-boggling numbers. And one of the reasons why, you know, we need to conserve invertebrates generally and the habitats that they live in is obviously they're a food source. And with organisations like the RSPB and other habitat management organisations, we could easily say that if we manage our invertebrates right and habitats for our invertebrates, then that's obviously going to be better for birds and everything else. So I think it's a question that we do need to raise. Should we be collecting insects? I strongly think we need to. Not everybody can do that. I certainly had at the FSC some people pull out of the projects we ran because they weren't prepared to kill things. Perfectly happy to photograph them, but they didn't want to they didn't want to take them. And if that's how you feel about it, that's absolutely fine. Certainly not going to judge you on that. But to have any detailed knowledge of entomology, you do have to capture invertebrates. Some of them are protected. They're protected from intentional killing, injury, or taking, sometimes protected from possession or control, or damage, destruction, or obstruction of access to any structure or place used by a scheduled animal. I'm not going to read through every part of the Act. You'll be relieved to know, but I think it's useful to have an understanding of which species and groups are protected and the level of protection they have. So the next few slides we'll go through quite quickly, but you'll get an idea of the range of species that are covered. So quite a few butterflies are covered in terms of their sale only, rather than taking them, as a list there. Likewise with these species, you'll notice on this slide the silver-studded blue, and then on the previous slide the large heath. Both of those you're not allowed to capture to sell. I remember receiving a phone call when I worked at Wixham Moss in Shropshire on the Welsh border, from a local landlady, and she said, "I think we have some of your butterflies flying around one of our bedrooms." And what had happened, a local chap away from the area had come in locally, and he'd been to the silver-studded blue site and collected a load of silver-studded blues. And he'd been to Wixham Moss and he'd collected a load of large heaths, and they'd escaped in his accommodation and were flying around. So people do collect these sorts of things and interact with them. So do be aware of that. There are a number of moths that have protection, where you would need a license to capture them. Likewise with a number of beetles. It's like stag beetle, they're forbidden to be sold. Hemitrum bugs, the New Forest cicada, which I think you'd do very, very well to find anyway. I don't think the New Forest cicada has been found in many, many years, but it would be illegal to collect one were you to find it. A number of crickets are also protected. Dragonflies, the Norfolk hawk, or the Norfolk Aeschina as it's called there, should probably be taken off that list if it already hasn't, because it's increased in numbers quite dramatically in modern years. But the southern damselfly is still pretty uncommon, restricted to a few habitats in the south of England and the south of Wales. Number of spiders, the fen raft spider, a lot of conservation work has occurred in recent years to do with that species. Similarly with the ladybird spider and the Dorset heaths, quite a lot of work going on there, and it's illegal to collect those. Mollusks, crustaceans and other invertebrates. Here are just a few of them, including things like the fairy shrimp, the medicinal leech and the freshwater pearl mussel, as well as the Roman snail. As with everything else, there are always exemptions. There are exemptions to do with any accidental damage or disturbance. Any of those species listed that potentially could cause serious damage to crops or livestock could be exempted. Anyone acting in the interests of animal health or public safety could be exempted. I'm not quite sure how that would apply to any of those creatures that we've looked at. And anything that's damaging or obstructing a place of shelter when it's inside a house would also exempt the removal, the collection and removal of those species. Carrying out scientific investigations and rescue operations or wildlife photography involving protected animals would also need a license from the appropriate authority. And then also protecting protected species in insect collections which were taken from the wild without contravening the law, for example, under license or before legal protection was enacted, then you're exempted from legal proceedings. There are a few European protected species we need to be aware of. The large blue butterfly being one of them. Fisher's estuarine moth, and then one that's escaped the slide. One of the ram's horn snails. And quite recently, there was a chap who was charged in court with capturing and collecting and killing large blues from one of the Cotswold sites. So very, very occasionally, there can be legal proceedings, but it's not generally common for that to happen, I think. So in summary, probably the majority of our rare wildlife isn't protected that much. And the chances of most people coming across those species in an everyday context of carrying out a survey are probably pretty small. And it's the same as those people who are involved in any sort of dodgy dealings or crime. They know their subject really well and they know where to look. So be aware that there is legislation, but it's relatively unlikely to impact on our day-to-day work as entomologists. I think it's probably all we need to say about that. And we'll move on to some slightly more common issues. And that's how planning has impacted on species of rare invertebrates. So there are a number of special areas of conservation that are strongholds of several of these species. Most of the southern damselfly populations are within special areas of conservation, likewise with a lot of marsh fritillary sites and the violet click beetle especially, also ram's horn snails and a range of other creatures. In terms of your local planning, attention should be paid to Section 41 species. For those of you who are not familiar with Section 41, it's basically species that were under the old UK BAP legislation, and back in the day when UK BAP was a thing, local authorities generally were really quite interested in protecting and looking after BAP species, and were able to fund quite a bit of work. Sadly, those days seem long gone. But in terms of planning, any Section 41 species should be flagged up. Anything that's within an IUCN threat category, so for instance, vulnerable, endangered, critically endangered, things that would have been in the old red data book for invertebrates from 1981, they're likely to be flagged up. And increasingly, the results of Pantheon invertebrate assemblages can be flagged up to planners. So if there's a particular invertebrate assemblage that's been found to be in favourable condition as judged against what it would be if it was a triple SI, then that may hold sway with planning. And generally local authorities these days have some form of chart or table that they go through that would hopefully highlight where rare species are. There's a code of conduct for collecting insects and other invertebrates published by the Invertebrate Link, Joint Committee for the Conservation of British Invertebrates. And again, I don't propose to go through this in any great detail. You can quite readily find it on the internet if you want to read through it all. But I've highlighted some of the more obvious sentiments that I think we need to pay attention to. And the first one there is no more specimens than are strictly required for a specific purpose should be captured or killed. Take one or two and leave the rest behind. Individuals of readily identified species, particularly butterflies, should not be killed where possible, nor removed from the wild unless required as voucher specimens or for scientific or educational study. These days, obviously, most people take a decent macro camera out with them, and that should do the job. Species that do not occur in abundance should not be taken year after year from the same locality. We'll come on to that a little bit later with an example, I think. But that makes sense. You go back year after year, or lots of different entomologists go to the same spot to collect the same thing year after year, and it could seriously impact the population of that species at that site. So we do need to be responsible. Taxa listed as being of conservation concern should not be collected except with the utmost restraint. That makes perfect sense to me. To know that it's of conservation concern is another matter, but as people get more experienced in entomology, generally your knowledge improves and you start to recognize things in the field sometimes that potentially could be of conservation concern. The collection of rare or local species from sites where they're already known to occur does not generally provide useful data and should be avoided except for the purpose of survey or other scientific study. Again, if you are looking for targeting rare species, maybe you have very good reasons to do so for scientific study. You don't need to take a voucher specimen every time in every case. At some occasions, you probably can't avoid that if it's a species that can't readily be identified in the field. But you should always try and avoid collecting rare stuff excessively. When obtaining early stages, such as leaf mines or galls or seed heads, never take all that can be found. That's sort of common sense, really. Leave as many as possible and allow the population to survive. Adequate biological records should always be kept, and this is something I'll come back to later. If we are collecting stuff in the field and identifying it, unless there's a very, very good reason not to, for instance, the landowner has asked us not to do that, then we should, if at all possible, share those biological records so that we pass the information on to recording schemes and others and contribute with the data. Collections should be properly housed so as to prevent deterioration or damage by pests. Again, if you've taken voucher specimens, it does actually pay to look after them. Some voucher specimens may well end up in a museum in the future. So do look after them and properly curate and house them. Okay. So what's the weirdest thing I've seen when I've been out surveying, I hear you ask? A Barbary macaque's bottom is what I would answer. I once did a survey in the monkey forest at Stoke-on-Trent, at Trentham Gardens, and the site is part of a site of special scientific interest, and we gained access to it with permission of the landowners. They hadn't really quite understood what we were going to do when we got there, and we turned up with sweep nets to sweep through the vegetation, and they said, "You're not bringing nets in here, surely?" And we said, "Yes, they're a standard bit of kit that we use." And they said, but the monkeys don't like them, because when we have to take the monkeys to the vets, we have to use nets like yours to catch them. And sure enough, the monkeys kicked off and weren't very impressed with us at all. So we had several people from the monkey forest around, making sure that we were safe and we were able to continue to do our job. Okay, so we're going to start talking about invertebrate field techniques now. So bearing in mind that this is part one of a two-part course where we're going to look at the use of analysing data using Pantheon, the first thing we need to discuss is how we come up with the requisite amount of field methodologies and visits to a site, and what methods we use to enable us to put together a list that is good enough to go into Pantheon. The slide in front of you has 4 columns. The one down the left-hand side that says F111 and F112, etc. That will probably be quite baffling to most people, particularly those who have not looked at Pantheon before. But what those codes represent are different types of specific habitat. I'll say no more than that for now until we come on to the next slide. And I want to show you all how to find out what they are. And certainly those of you who are coming back next time, it would really, really help if you did that. The second column represents the different types of invertebrates that the methodologies will find. So col is Coleoptera, het. It represents the bugs, the tree bugs, Diptera flies, Hymenoptera, Lepidoptera, and that probably goes through that list there. So what that list is saying is that in a particular type of habitat, to enable us to test the assemblages through Pantheon, we need to look for beetles and bugs by ground searching in late April and May, and we can also pitfall trap at the same time. In, again, that particular habitat, the first one, the F111, if we want to sweep for flies in spring and summer, that will help us on our way towards achieving favorability in terms of the Pantheon software analysis. And then likewise, flies and Culeate Hymenoptera, we can spot sweep, i.e. sweep where we see things in spring and summer. And so each of those habitats will have their own recommended methodology involving the type of species that we search for and when we do it and how we do it. And we'll have quite a big chunk of this talk will be looking at those methods, and you'll be glad to know we'll soon be looking at pictures and photographs rather than very, very text heavy slides like the first 24 have been. So please don't worry too much about Pantheon at the moment. It's just a matter of understanding the groups of invertebrates we're searching for, when we're looking for them, and how are we going to catch them. So, and anybody who is planning to join us for the Pantheon webinar on the 10th, I think, is it on the 10th? Something like that. So we have, so those codes that we just saw in the last slide are called SATs, specific assemblage types. And to look up what the SATs mean and what they're about, unfortunately, Pantheon doesn't have a very easy way of getting to that information, but there is a way, and here it is for anyone who's interested to have a go. You'll need to sign into Pantheon first, so create an account, which is very, very straightforward to do. And once you're in, you can go to the help button at the top of the page and then click on the glossary and this is a sort of representation of what comes up: a title, a summary, and a tag. And if you scroll down the tags until you see the word sat, then click on the sat, and that takes you to a special page that explains all the different species assemblage types. And it will tell you what type of habitat it is. In this case, the one I've circled is part of the bare sand and chalk sat. It will show you some photographs of those sorts of habitats. It will tell you a little bit about the invertebrates associated with those sorts of sats, tell you what the target is, and a bit more information. And this is something that we'll be covering in the next webinar. So I'll leave that up there for another 30 seconds if you want to screenshot that, or we can send it to anybody who doesn't get it during the talk, who wants it later. So well, set yourself up a Pantheon account first. To get to Pantheon, it's really easy. If you Google Pantheon BRC, you will get to it very, very readily. Get yourself an account, help, glossary, scroll down the tags to SAT, click on the link, and you'll get through to all the information that you need in time for the next webinar. Right, we'll move on. So there wasn't always Pantheon, and entomologists have been carrying out surveys for many, many years. This little sort of snatch of a table that actually occurs in, I think, one of the reports from somebody who's on the call this evening. Thank you, Martin. Gives you a good example of how an entomologist would set about defining the different types of methods of finding invertebrates that we're going to talk about in the next part of the webinar. So just sort of reading down that list. So all those site compartments are in the middle table, obviously the dates on the left, and 10 minutes of sweeping and general recording were done at one part of the site. General recording was done various other places. Beating and sweeping was done along the hedge. General recording at another part of the site. Pitfall traps were put in at a number of other places. Beating and sweeping and pitfall trapping. So you can see there's an array of methods that you can use, and as long as they're sort of balanced, then that's absolutely great. Having an understanding of habitats and methods will help that, and we'll come on to that shortly. In fact, now. So this is me on the left in my Natural England Field Unit days a few years ago. And I'm sort of kitted up with the equipment that I generally take out into the field for most of the work that I do when I'm looking for the species I look for, which are mostly flies and bugs and a few other bits and pieces. When I worked in the field unit, I worked alongside other entomologists who had other specialist skills, be they Aculeate Hymenoptera, spiders, mollusks, and a variety of other things. And I think that's what you'll find if you don't have any expertise of entomology, or a very little bit. It's unlikely that you're going to be able to collect enough, or certainly identify enough species to be able to test through Pantheon. You do really need a few people who are good at different groups of invertebrates. So beetles, flies, bugs, bees, wasps, and ants, maybe Orthoptera, maybe Lepidoptera, maybe spiders. So I've got my sweep net, I have my pooter, I have a hand lens around my neck, and I have a pot in which I have alcohol in it, which we'll come on to a little bit later. And probably 75% of my entomology is done in that way as part of a wider team. And as an action shot, sweeping the net around, you do get quite well developed arms and wrists when you're sweeping a net around all day. But it's a really good methodology. You get right down into vegetation and you get all sorts of stuff. You have to be a little bit careful if you're working around others. I've seen so many people put their net pole under their arm so it sticks out the back, and they turn round and it looks a little bit like something out of a Laurel and Hardy film, and where you really have to avoid striking somebody else. The other thing you need to be careful of is if you've swept and potentially you have something like a social wasp in your net, you don't really want to put your head in if there's a net. And I didn't comment about that on the last slide. There's my colleague with a head in a net, emptying the catch of flies and other flying invertebrates, or in some cases unflying invertebrates, beetles and not beetles, a range of other things that could end up in there, and transferring them over to a collecting pot. There's an awful lot you can achieve by ground searching. There's a couple of different methods here. My colleague on the left is using a pooter to poot up spiders. She's disturbing the vegetation and seeing what scuttles away, and then being very, very quick and pooting up the catch. On the right-hand side, another colleague is sieving a leaf litter and plant debris, and sieving it into a white tray. And then she has a really handy pair of goggles on that magnify what's in the tray, and she would be collecting probably mostly tiny mollusks whilst doing that. So there's an awful lot that can be gained by properly having a look in your surroundings, getting on your hands and knees, sitting down, making yourself comfortable and seeing what you can find. In similar circumstances, here's some of the team that I used to work with examining a riverbank, really stony, gravelly riverbank, looking for beetles and spiders and a variety of other things. Turning rocks over, certainly there'll be a bit of sieving going on. A couple of people have pooters, and a bit of time doing that can be pretty productive. This is called a Malice Trap. Malice with a capital M, because it's named after somebody, after Dr. René Malice, who was a Swedish scientist. And this is basically a flight interception trap. So here you can see it's situated at a sort of scrubby edge of a bog on sphagnum moss. And so your catch, in this case flying insects, would fly into the black bits of the trap in the center, and then start to walk up towards the light. And you may be able to see at the top left-hand part of the trap, there's a collecting vessel, and that's filled with, in this case, it was filled with alcohol. In the old days, you used to collect dry samples in Malice traps, and you can leave that out for as long as you want, and change the bottle of the trap every few weeks maybe, and it will sample in your absence. So it does collect an awful lot of stuff, and this is something you need to be aware of, thinking back to the code for insect collecting. If there's something particularly rare there, this could easily collect all the population. So you would only Malice trap for a short period of time, maybe a day, maybe a few days, not at all, certainly for weeks and weeks, if you had any concerns about that. Malice traps, like most of the forms of entomology in terms of the field work, are very, very effective. And you will get many, many thousands of specimens using this methodology. And it's very, very easy to find invertebrates, but it takes a long, long time to identify them. So you do need to bear that in mind also. Light traps. Light traps are obviously the entomologist's friend. They're going to bring in moths, but they're going to bring in a whole range of other things. I often would go and stand by light traps and not look for moths, but find crane flies that have been attracted to them. Caddis flies, beetles, all manner of other things turn up at moth traps. So they can be a really useful contribution to getting a site list or to part of the survey work that you're carrying out. There's a lot of invertebrates can be found by looking in dead wood, old trees, veteran trees, dead bits of young trees, all sorts of situations. Here a colleague has gone into a tree, not that tree on the left, but a similar one, and pulled out some rotting wood with a trowel, and she's sort of picking her way through it and sort of demonstrating it to other colleagues. So particularly if you're interested in the beetle assemblages of your site, a bit of time going through dead wood will certainly help towards the realization of that. Likewise, here we had some fairly elderly wood that had been stacked and started to sort of decompose, and a few of us spent a bit of time going through this, taking bits of bark off. That looks as if it's been quite destructive, but I can promise you that it wasn't. It was pretty much like that when we found it, the result of lots of limbs from trees being sort of moved and stacked and then taken away. And we found a number of deadwood species, longhorn beetles especially, in that sort of habitat. If deadwood is your focus, then you may want to use vane traps. These are plastic traps. Again, they act as interception traps. And you can see here they've been put up in relatively old trees, or trees with cavities. Both of these are different oaks on a site actually quite close. In fact, it was the same site as I saw the monkey's bottoms at in Stoke-on-Trent, and they were put up in April, and they were left there until October. But every 2 weeks, the chap there up the ladder would go and change the bottle at the bottom of the trap. So beetles and other things, flies, a few other bits and pieces will fly into the plastic, and then they'll get knocked down into the funnel, and gradually they'll find their way going further down into the collecting bottle. It's a very, very good method for deadwood invertebrates. Only recently been employed sort of more widespread, but it does work incredibly well. And a list that you've collected using vane traps on a site like that will go a long way in terms of being able to properly assess the deadwood fauna through Pantheon. Here we've got a very tussocky, grassy heathland site, and there are a couple of different methods you can employ here. One is the same sort of hand searching as was demonstrated early on, The other thing you could do here is do some tusking. So you could get a relatively sharp saw and cut into one of the tussocks and shake it into the white tray that the colleague has there and see what's in there. It's pretty good survey technique for beetles and a variety of other things that tend to tuck themselves away and aren't necessarily that obvious. You can also maybe make out in the photograph that he's got a glove on. Again, we have to be aware of health and safety in all the work we do. And this is a Heathland site. It certainly has had adder recorded on it before. And the last thing you want to do is separate a tussock and find your hands in contact with a snake. So you always have to be a little bit careful when you're out in the field. Pitfall traps, a mainstay of entomological recording. Again, these can be used in lots of different ways. They can be put out as individuals, like this one here in the photograph. They can be put together in numbers. We used to have 1 meter square of wire and put half a dozen traps underneath that wire. The wire basically helps prevent things getting in the pitfall trap that shouldn't be in the pitfall traps. In the photographs there, the photograph on the left has a liquid in it, so that I would suggest that pitfall trap has been set to stay there for a little while. So it has a liquid in it, possibly antifreeze in that case. We sort of use a slightly less harmful chemical these days, which I'll come on to in a little while, but that would act as a preservative, and any insect that fell in there would sit at the bottom of that, and it wouldn't rot. You don't have to leave pitfall traps in for days or weeks. You can just leave them overnight, like the one on the right hand side. So that has just been set, left overnight. Ground beetles, spiders, variety of other things may well fall in during the night, and then you would go around the next day and collect them. And if you needed to take them, you could pop them in alcohol. If you are able to identify what there is, then you can let them go again. But it gives you that bit of flexibility. Some of the machinery you can use. This is a DVAC, a vacuum sampler. This was sort of really the first generation of sampler before the more recent ones. This had a big petrol engine on the top of it, and was very heavy and very noisy and not entirely pleasant to use. Thankfully, these days there are a range of vacuum samplers that have batteries on them and are very light and portable. The only difference really is this one, you could use it for most of the day if you could stand lugging it about, whereas the battery powered ones don't last very long. But it depends what sort of use you're putting them to and how long, and the sort of size of the site. This particular site you can see here is quite a big one. It's salt marsh at Morecambe Bay, and so I spent quite a bit of time wandering around tussocky areas, sucking up all sorts of stuff, spiders and bugs, flies, and all sorts of things. So they come in really handy. Here we've got a range of people doing a range of entomological techniques. You can see that they've recently had the vacuum sampler going. There it is on the left hand side. It's a smaller one. It's still a petrol one in this case, but certainly a lot smaller than the DVAC that I was using in the previous photo. We have our spider person and a coleopterist in the background who are fertling around in vegetation and turning rocks over at the side of streams there. And we have our mollusk person in the near ground. She will have again, either sieved or brought some material into the tray and is searching through it for her group of animals. So one of the things that we have to bear in mind is, what are we going to do with all these things we're catching in the field that we're sweeping and we are sucking up with vacuum samplers, or we're fertiling for, or we're pitfalling? And there are a number of chemicals that potentially we could use. We tend to use propylene glycol these days in our vane traps and in our pitfall traps. This is a food grade propylene glycol, so it offers very little risk to anybody using it or coming into contact with it, as opposed to some of the more nasty chemicals like antifreeze, which is really quite poisonous. So that's one of the chemicals I use. The one in the middle is IMS, industrialized methylized spirits, a type of alcohol. This is added 30% distilled water, so it's 70% IMS, and that preserves most of the inverts that I capture these days. We tend not to have time when we're on survey work to keep things dry, because we're going from one site to the next, day after day after day. We just don't have the time to do that. So everything goes in alcohol, like the picture of the right on the right of the pot, and that's how things stay for us until the winter, when we start to sort our catch out. So propylene glycol and IMS. The third chemical there on the right hand side is a little bottle of ethyl acetate, and that is a sort of knockdown chemical that if you introduce it into a glass tube where you have an insect, it will very, very quickly kill it. And a lot of entomologists use that. I tend not to use it that much, because I find that if we're doing this day after day, I don't want ethyl acetate fumes around me all the time. And I tended to, before I used alcohol a lot, if I took stuff away, either still alive from a site in a pot or what have you, I would put it in the freezer overnight to kill it, rather than use a chemical. So a lot of people still do use ethyl acetate or chemicals very similar to them. I tend not to these days because of the frequency of usage. And I think it can probably do you quite a bit of damage if you're inhaling that in a pewter or a tube. So something we need to bear in mind. And this is the result of a number of surveys in a season. You will have lots and lots and lots of specimens if you're going to survey a site. And you have to start to sort of think about the scale of that and the reality of getting these things identified. If you are in a situation where you really know you're not going to be able to do that, then is this for you? But fortunately, where the surveys I've been involved with, as I say, I work with a number of people, and what we tend to do, we collect all sorts of different material, and then the flies would come to me, and I would give the beetles to the coleopterist, and the culeate hymenoptera to the hymenopterist, and that's how we would go about doing our survey work. But don't underestimate the amount of time it takes to identify things if you haven't looked at them before. Fortunately, there are some great initiatives showing you how to identify things, particularly through Tanyptera and other similar organisations who do similar work. It's probably never been a better time to get into invertebrates if you want to. There's plenty of opportunity there. Right, let's look at some sites and talk our way through how we would approach survey. So the next several slides are all similar to this one, different types of habitat, and we'll look at some of the features. Where might we survey? What are the key features in each photograph? So where would we spare our time to do survey work? So this is a heathland site in the West Midlands. If you sort of scan your eyes around it, there are some interesting bits of habitat. Probably first and foremost, and what leaps out to me is the sandy bank, the open sandy bank with lovely friable sand that is more or less south facing. So that's going to be really wonderful for Aculeate Hymenoptera. So there's going to be lots of bees and wasps breeding in that area. There could well be some flies that are parasites of them around there. We've got lots of flowers nearby, so it's very likely that those bees and wasps are going to be taking nectar and pollen from some of those plants. We've got a bit of heathland beyond it. There's a sort of slight indication there that there might be some wetter ground occasionally, depending on the conditions. There's some scrubby stuff beyond that and some other grassland. Woodland edge is another sort of interesting feature, and you've got a nice sort of situation in the distance where the scrub is sort of blending into larger trees. So there's a whole range of potential habitats that you could spend time at there. And seeing something like that, you really wouldn't want to give that a miss if you were in any way serious about assessing the heathland. When you get to know Pantheon will say that the assemblage or a couple of assemblages are most likely there, and we'll go into that next time. And you would be aware of the main groups of invertebrates that would be of interest there. So bees and wasps, flies, beetles, and they're probably the main groups that are represented there. This is a different habitat from this spring. This is the northwest of England. This is a woodland trough site, and one of the key things there you can see is the scrub and the woodland edge, and those habitats from about that time of year onwards become really, really important. There'll be a range of things using the flowering blackthorn to nectar on, and because of the way the woodland and scrub edge is aligned, the right hand of the picture is south facing, so that south facing edge will get warmer than the surrounding land, and would be a good place to sweep along to spot search for invertebrates nectaring, or on leaves, or whatever you are after. So south-facing, warm, scrubby woodland edges are always going to be pretty rich once the season gets going. So that's where you'd target. You'd probably get very little from that grassland in the middle in comparison. This is a river in the Welsh Marches, just around the sort of English border. It's a river that has quite a lot of tufa formation on it. There are going to be all sorts of little seepages coming into the river. You can see there's a lot of bryophytes growing on the tufa. There's fallen dead wood on the left-hand side, which will be rotting down and providing habitat. On the right-hand side, there are smaller bits of timber that have fallen in to the edge of the stream and could be moved by the water, forming little log jams in various places along that stretch of river. And so that offers up all sorts of opportunities for, particularly for flies. It's a very, very sort of rich habitat. And if we were to look, as we will next time, at the assemblage associated with that sort of habitat, it's going to be very, very fly heavy. And that sort of begs the point. It's handy to know that beforehand, because if you come to a site like this and decide that you're going to survey it for beetles, then you may find beetles, you may find plenty of them, but it's not going to do a lot for your chances of getting an assemblage out of your species list. So you very much need to know what you're looking for, and we'll talk more about that next time. Rather wonderful calcareous meadow. This is down in Hertfordshire, I think. Really beautiful floristically, some interesting plants there, and in terms of the invertebrates, a whole range of things, lots of butterflies, lots of Orthoptera, lots of soldier beetles and the like. Really rich bit of habitat. Not a lot of scrub there, and one of the issues we had with the management of this site was there was a very active friends group who removed scrub as soon as it showed its face on the site, which in some cases is great. You wouldn't want a meadow like that completely dominated by scrub, but you certainly would benefit from some scrub in some parts of it. And when we surveyed the site and looked at some of the previous surveys that had been done there and put the data through Pantheon, there were several species that showed that connection with either woodland edge or bits of scrub, and those were the recommendations we made, that we could cope with maybe 10% of scrub in certain parts of the site. So flowery habitats like that are always going to be a joy to survey. You'll get loads of aculeate Hymenoptera and a range of other things that represent the pollinator assemblage. Very nice. This is a site in the Brecks, part of the Brecks Arable Margin project that I've been involved with. You can see on the left that the fields there are actually part of a pig operation, and the farmer in this case gets paid to plough the margins once a year, and the resultant botany that comes up is pretty special, and the associated invertebrates also are pretty special. So that the sort of strip in the middle where the person is kneeling down is far richer than the bit on the right hand side that's slightly more sort of scrubby, that doesn't get ploughed. Although you get probably more commoner invertebrates on the right hand side, it's where the rarities are down the middle that are associated with the bare ground. So that's the bit of interest there for us. The trees around the edge, not so much so at all. It's those arable margins with their really friable sandy soils that are going to produce the interesting things. So here we've got a local woodland close to where I live, in its first sort of rather belated lime green burst of spring. Mostly birch trees, a few older trees associated fairly close to the ditch that runs through the middle of that shot, and a few more mature oaks around the edge. So this is a site that when we get to July, it's mostly dominated by bracken, so it's quite difficult to survey. So we would probably want to get our visits in a little bit earlier in the year, catch the spring stuff, catch the ditches before they get inaccessible. Fortunately, this site has undergone a bit of habitat management recently, and they've blocked some of the ditches to try and retain water for longer. This would have been at one point a bog, like a lot of the surrounding landscape, but it was drained for agriculture probably back in the 1930s or something like that, or around the war. And as a result, dried out and dries out still very, very quickly. But nevertheless, probably quite good for invertebrates at times and in parts. And there's plenty of dead wood around the site that might be of interest. You get quite a lot of fungi growing in autumn on the trees, which could house some interesting beetles. And the ditch that runs through the middle there and other ditches around the site certainly will be of interest for a whole range of stuff. Probably a bit of classic habitat here. This is a bit of valley mire from the New Forest. The very fact that it's in the New Forest means that it could house all sorts of really quite interesting and rare species. So we've got a range of habitats going on here. We've got the valley mire itself, got the wet bits. Then we've got slightly drier areas either side. You can see that some of it's bracken dominated as it goes up the slope. I've got a range of scrubby trees and gorse in the distance, and slightly more dominant woodland in the far distance. So quite a lot going on here. There's a little bit of dead wood I can see on the right hand side. So it's a matter of picking the best bits that you think you should survey here. And the wet bits, the scrubby bits, the woodland edge, and sort of any heathland that isn't bracken dominated are probably going to be the areas we choose there. This is another site from the West Midlands. It's actually a brownfield site. It's formerly an old colliery, and it's been not necessarily rewild, but certainly turned back to nature. Here you've got an area of wetland in front of us, which goes through sort of most of the middle of the shot. Quite close to us in the shot, we've got areas of heathland, and within that we've got areas of bilberry, quite nice heather. In the distance and sort of behind the trees on the right hand side, we've got an old colliery tip which has been capped, but it still has quite a lot of seepages, interesting seepages that come out of it. And then further round to the right, as we go out of shot, there's heathy grassland with bird's foot trefoil that we know has housed dingy skipper butterfly. So quite a range of habitats here, as well as the woodland on the sort of far left. So we may have to use a range of different techniques to adequately survey this. And these are the sorts of things we would want to bear in mind before we started. How long are we going to spend on each habitat? What are we going to get out of it, depending on where we are in the country? There are a number of different habitats there. How do we go about doing it? All things we need to think about. Another different habitat again, this is one of the Dorset heaths. And you can see that it grades down from a scrubby bit at the top. There are areas that have been cut, and then we grade gradually into salt marsh. So again, very, very different habitats as you progress through that shot into the distance. And as a consequence, the invertebrate assemblages associated with those different habitats are again different. So you would need to have a number of different strategies if you were wanting to take all that into account and survey it all. Lovely bit of habitat, probably not too far away from where the aforementioned ladybird spiders were introduced, as it happens. Floodplain grassland, another habitat again. This is down in West Sussex, close to Pulborough, and there are a number of of ditches that run across the site. Several of those ditches house really quite important populations of mollusks. There's a dragonfly assemblage associated with this site. There are really nice bits of lush vegetation at the side of the ditches that are rich with a whole range of things, beetles, soldier flies, a variety of things. And then we go into sort of slightly drier grassland once we get away from the ditches. Every now and again, the whole thing floods and sort of deposits mud on the site, and how sort of rich and verdant the vegetation is. So again, there are a number of different strategies we'd have to employ if we were going to survey everything. If we're going to look at the ditches, we need to be in the ditches with pond nets. If we're doing the terrestrial stuff around the edges, then we need to be sweeping those river sides in a safe manner. And if we're looking at the dragonflies, then I think probably we need to do a bit of both. We probably need to get in the water again and look for dragonfly larvae. We need to be looking at the edges for shed exuviae, as well as looking for adults. So a whole range of things we could do there if that was our site. Probably my favourite bit of habitat that I've worked on is areas like this, which are, in this case, this is on the Isle of Wight. It's soft coastal rocks. They have been eroding and shifting, sometimes sort of, slumping, and that leads to a very, very quick turnover of habitat, lots of bare ground, lots of wet areas, seepages. So in the photograph, you can see areas of sort of pond vegetation there, just on the right-hand side, and some of them get moved in situ as the cliff slumps. So probably not the easiest site to work. You have to be very, very aware of health and safety issues on these sites, and you probably won't find masses of invertebrates on some of the bare ground, but what you do find will be unusual and quite interesting. Fabulous place to work. This is a cut over peat bog in Shropshire. You can see that many, many years ago, this was cut and emptied of peat with a few little bits left. And then over time, the ditches have been blocked, water has been kept in, and the sphagnum moss has started to recolonize this area. And probably in another 20 years, you probably won't see much else other than a few tussocks sticking up, while the rest of it is sphagnum moss. Particularly good for a range of things, range of uncommon things, spiders and flies and beetles, etc. Can be quite difficult to survey sites like this, again in terms of safety because of the depth of the ditches, but it's certainly going to hold a whole range of invertebrates. Now, the chances that you will be involved surveying on your site with somewhere that's an old cut over peat bog is probably quite low, but if you ever get the chance to, they're wonderful places to visit. And I know certainly Tanyptera has been involved in its fair share of work on the Cheshire meres and mosses. Coastal headlands. My brand new hobby is walking the Wales coastal path, which I'm doing at weekends. And I'm seeing lots of habitat like this. This bit is a triple SI for its botany. Once you get past the gorse, it opens up into rather fantastic calcareous vegetation. But you get a lot of coastal headlands like this that could be, on the face of it, quite difficult to survey, very, very breezy, but with bits of shelter. It's always going to be in the bits of shelter where you find probably the most things of interest. These sites are very, very difficult to run through Pantheon when you're just surveying a very narrow strip of land over a sort of a long period. And we've done similar work to this. This is in Wales, but we've done work to sites like that in Cornwall, and they can be really, really difficult to survey. But we have to do our best. And then perhaps a piece of not so good habitat that occasionally we would probably get asked to survey. This is a grouse moor in, where is it? I think it's Derbyshire. It's the Cheshire Derbyshire border, I think. And you can see that it's cut regularly to have fire breaks in there. Any areas of wetland will have been drained, and it's not a particularly friendly place to survey. I have, as I say, I have surveyed things like this, and invariably they turn out to be unfavourable in that sort of state. So not everywhere you get to survey will be an absolute joy. In fact, probably most surveys that people will end up doing will be slightly substandard sites, but generally that's how it works. And we have to just do our best. We approach it in the same way. We look for the best bits, we look for different types of habitat, and we have a think about the methods that we looked at and how we would go about surveying on the site that's in front of us to the best of our ability. I mentioned earlier that things are changing. In terms of taxonomy, DNA is certainly, DNA-based monitoring is starting to gain ground. I was made aware of this little diet analysis of Bechstein's bats that was carried out in Oxfordshire, Buckinghamshire, Burnwood Forest. And they obviously collected Bechstein bat excreta and DNA analysed it and came up with a list of invertebrates that the bats had been preying on. This came out with some quite interesting results. It showed that the DNA technology wasn't there yet to properly be able to have a full analysis. For some species, it couldn't identify what they were, or it could only identify them to order or genus in some case. And then some of the species that it analysed and came out as species could be said to be questionable. There was one fly particularly that had never been found in Oxfordshire or Buckinghamshire, and the habitats within the woodland and around the areas that the Bechstein bats would have flown around was certainly not the habitat that fly occurs in. Now, of course, the fly, it could be right. The fly could have found its way into that habitat, migrating or moving or being carried by the wind, or a number of other sort of reasons. Or it could be that the analysis was wrong in this case and it was a similar species. And that's sort of where we are with DNA. It will only get better though, and it's something as entomologists we do need to take into account. I think we all hope that the days of the taxonomist who uses traditional methods is always going to be needed. And probably the way to look at the DNA stuff is that it should assist us rather than hinder us. There's a program at the moment being run by the Natural History Museum called the Darwin Tree of Life project. And the idea ultimately for that is to DNA barcode fully every species that occurs in Britain. That's a big project, and it's something at Natural England we've started to get involved with. Some of you may have been part of that. We had an event a couple of years ago, actually in the olden days before COVID, up at Ainsdale NNR, and entomologists from the Natural History Museum and Natural England and various other places. Lots of local volunteers got involved, helped out by collecting invertebrates and starting to put them through a particular system that the NHM were using. And the idea there is we try and fill the gaps and also double check the existing species, the species that we currently have barcodes for as well. So there's only going to be more of that work carried out. So I think we probably have to be aware of that. Just a couple of slides to go now. So we have done our work. We've seen our site. We've decided how we're going to survey it. We've had to think about which methods we're going to use. We may want to Malice trap it, do some pitfall trapping, vacuum sampling, all those different methods. We've curated everything, pinned everything, identified everything, and this is what we end up with. A couple of pages of species, and the next talk that I do in a few days' time, next week, whenever it is, we'll start with these 2 slides so that we discover what we do once we've got the information. But to finish with, I want to re-emphasize how important it is that if people are going out and capturing invertebrates, identifying them, then really we do need to record that information. And one of the best ways of doing that is iRecord. There are lots of opportunities. If you've never heard of iRecord, there are lots of opportunities to get to know how to use it. And it's basically the best way of getting your data usable by recording schemes and people who make decisions in terms of nature conservation. So please do use it. I think I'm at an end.
Pete, that was excellent. Thank you very much indeed. Yeah, you were really clear, and you have a way of speaking that I could really listen to you all night. You really came across extremely well. We've done a lot of webinars over the past year on identification, but this is a really entomology, survey techniques is a real fundamental and something we haven't covered, and I think this is really important to have someone like yourself with your experience go through it and cover it really well. And I'm very much looking forward to next week. I really enjoyed your sort of journey through just so many different invertebrate habitats there that people will now go out and recognize. And I think it was also I think it was very understandable for those just getting into invertebrates too, and that's brilliant. So I think we'll go into some questions now, if that's okay, Pete.
Yeah, of course.
Okay, so the first question that we had through was from Antonella Calazo. I'm sorry if I'm pronouncing that wrong. What university or how do you recommend the start of education before working in the field? I am considering this field of research in the near future.
Oh, fantastic, Antonella, that's great. I suppose that rather depends where you are. There aren't that many universities that run entomological degrees, but certainly in the UK, we have go on, Gary, help me out. We have the MSc at Harper Adams, which is a really good qualification.
I think that's the only master's degree in entomology in the country.
Yeah.
And then probably the FSc MMU biological recording
Yeah,
MSCs.
which is the route I took. I, basically took the decision quite early on to get as much experience as I could, and to try and back that up with a qualification. I suppose that was a different era than today, so I'm sort of less knowledgeable about how you would go about that today. But I suspect there are many universities who, around the world, who would run environmental science degrees and a variety of others. But unfortunately, as I say, I'm a little long in the tooth and too far away from that world these days to be up to date on it. Maybe anyone else can chip in.
Yeah, I think there are probably lots of university courses like conservation biology, zoology degrees, where you can probably have the opportunity over the years of the degree to really specialize through doing your thesis or undergraduate degree, doing your project, and just choosing the modules that are most going to be closely related to doing actual field work and getting into identification. But I think real specialist courses are quite few and far between, but I'm not that up to speed with it. Don't know if anyone else has. Yeah, it's just, yeah, Harper Adams also do a zoology with entomology BSc. Okay, so you can do entomology as part of a degree at degree level, not just a master's level. Thank you, Alex. But yeah, if anyone else wants to chip in on that and help out Antonella, then just maybe put something in the chat if you know of particular courses out there. Okay, I'll go on to another question now. This one from Steve Docker. Hi, Pete. Are there any technologies that can help with species identification and reducing the problem of ID expertise? DNA techniques, perhaps?
Yeah, I think we sort of covered that at the end. It's very much a new and emerging field. I don't know how. Maybe is anyone doing any work on the museum collections to to barcode stuff at Liverpool? I don't know. Are they?
Well, we do now have the facilities to do the first bit before you send it off to the sequencing, to do the PCR work. But we haven't done a great deal yet, actually. Through the pandemic, we're not allowed in, so we just got going with it before. So we'll see. But there's certainly. I think it's much more effective in terms of doing barcoding on fresh material, which is not a great strength of museums, and also for material in alcohol. IMS is not very good for preserving DNA, and that's what most of our collections are. But in terms of reducing the problem of ID expertise, I think recognition to certain levels using photographically is coming on, isn't it? The technologies in that way, but I don't know what the latest is. That
seems to be very, very good. Some of the stuff that's been developed. I know Stuart Ball has done a lot of work on hoverfly ID recognition from photographs from people that submit stuff to their recording scheme, and that's getting a pretty good hit rate. And again, that's only going to get better. The more artificial intelligence that's used. I think ultimately the question to this is if I'm alive to see it in 30 years' time, you know, will entomology be the same as it is now, or it was 30 years ago when I started? It would be really interesting to see what technologies have developed. You know, when I was starting, there was always, you know, someone said, oh yeah, in 30 years' time, you'll have a device that enables you to identify everything. You'll just point it and it'll tell you what it is. And we're not quite there yet, but we're on that journey. We're certainly on that journey for some things that are different enough for the changes to be recognised by artificial intelligence. And that's the difference. It's the pattern recognition. But there are a lot of things where it's still going to come down to good old-fashioned sort of, you know, taxonomy to sort of QA it.
Yeah, and I think that for 30 years ago, I think the getting that expertise is probably more difficult than it would be today with much better, you know, lots of modern keys, and experts at hand on Facebook groups and all sorts of different types of key as well, you know, that the FSC have developed. I can't remember what they're called now, but I think there's the problem of idea expertise is probably greater in decades gone by, whereas there's just more aids now, and if photo recognition isn't perfect, at least it can get you close, and then you're that speed things up to actually check it and make sure of what it might be. Okay, well, we'll go on to another question. Thanks, Steve. Another one from Antonella Colazzo. How common is the career of forensic entomology in your country? Have you been asked to share your expertise in court before?
No, I've not. That's a very, very specialised part of entomology. I don't know how many forensic entomologists there are in the UK, but yeah, it's largely down to the study of quite a small group of species, and knowing intimately the lifestyles and when the instars of each of those particular species and what they feed on. I've read some interesting books about forensic entomology, but it's not something I've been involved with at all, and I would be very surprised if I'm ever called to court, at least for that. But thanks very much, Antonella, for the question. I can see the next couple of questions about propylene glycol. So propylene glycol is okay in plastic bottles. It comes in plastic bottles, and we put it in vane traps, which are plastic. The thing you don't want to put in plastic bottles is ethyl acetate. It's quite easy to get the names mixed up. Ethyl acetate will melt plastic, and so you don't want to put it in plastic bottles or tubes. In terms of how you process stuff for mounting, it's a combination of putting in alcohol and distilled water. Obviously, if you're looking at hard-bodied creatures like beetles, they're much easier to process and get them ready for mounting. If you're looking at processing soft-bodied creatures like flies or aculeate Hymenoptera, where drying them out is essential to see the patterns or hairs on them, that's much, much harder, and probably wouldn't even contemplate trying to sort flies out from propylene glycol. So most of the invertebrates that they're likely to capture as you're using them in pitfall traps and vane traps are going to be things like beetles and spiders rather than things like flies. So yeah, beetles, no problem. Spiders would be kept in alcohol anyway, so that's not not a problem. Anything that you need to dry would be much harder to process, and it's probably not worth trying.
Okay, thank you, Pete. Yeah, I think propylene glycol is, so it is, as you said, it's food grade, and I think it's used as a food additive. So the volatility of it is very low if you sort of look up the chemical.
Yeah.
So it's much better. I think it was much more common, Pete, wasn't it, to use antifreeze in the past?
Yeah.
In traps, which is which is much more potentially harmful to wild animals and dogs and things. Discover them. So I think this is I think it's only quite a modern thing to use propylene glycol in entomology. So, but I think it's a very good move. So there's a question from Angela Price. Do you ever find tardigrades in wetter areas like sphagnum?
Tardigrades are very cool things. I'm sure if I look for them, I would find them, but because we don't use them in the Pantheon analysis, it's not the sort of thing I look for. However, I've done some work on soil sampling with Matthew Shepherd, who's our Natural England soil specialist, and he's got he always finds tardigrades and takes great delight in showing them to fellow surveyors or people in his class. And he's got a big projector that sort of scans it up onto full size of the wall, and they're wonderful things. So brilliant question. Thanks, Angela.
Thank you. Okay, a question from Ian Strand. Can sweep netting be standardized for monitoring? Is there any evidence on repeatability?
Yeah, it used to be standardized in common standard monitoring, and you would undertake a certain number of sweeps, 50 sweeps, 100 sweeps, or whatever. But how standardized that is, I'm really not certain. In terms of how we would survey in Natural England, we might only visit the site every decade, and can you guarantee that sweep netting was done in the same part of the site by the in the same way as the last person did it? It can make a big difference how you sweep. So I don't think there's any evidence that you could repeat it that closely, but that's how it certainly used to be done. A lot of the work we do now is standardized at a sort of different level. So we would do 3 visits that cover 3 days with a number of different methodologies depending on those that were most appropriate to the habitat, rather than go in and do 50 sweeps in this part of the site. So yeah, I think it's difficult to show evidence on the repeatability if you're not doing it very regularly.
Thank you, Pete. Yeah, very good question. So
yeah, absolutely.
Okay, question from Naya Howells, and again, I apologies for pronunciation. Thanks, Pete. What do you think about using bioacoustics as a standardized technique for surveying inverts like Orthoptera, especially for more biodiverse regions like rainforests?
I think in this country we're a long way behind on bioacoustics, and we're just starting to have discussions in Natural England now about how we might use bioacoustics more. I think more tropical parts of the world, like Naya suggests, are ahead of us on this, and a lot of the research that we're reading currently is from parts of the Australian rainforest around Queensland and places like that, where where they're much more used to carrying out those sorts of surveys. But there's definitely a groundswell of interest in bioacoustics and how it might alter the way that we survey. So I absolutely agree that I think this is definitely going to be something that grows in this country as we become more familiar with the techniques and what it can show us.
Yeah, I think it's a great sort of topic for development, really. But there's on a very basic level, I know on the Manchester mosses here in the northwest of England, they sort of do transects with bat detectors to count the bush crickets on the mosses. But of course, we don't have many orthopterans here, do we? In the UK.
I remember one of the first ever entomological jobs that I got to do as a consultant was to go and identify whether a colony of Rose's bush cricket was still at a site at Fleetwood in northwest England. This was when they were pretty uncommon in this part of the world. And I arrived on site, I opened my car door, I could hear them so clearly. I closed my car door and I drove away. And I'd answered the question, are they still there? Yes, they are. And it was the easiest 200 quid I ever made. But yeah, definitely it's been a little bit facetious, I know, but certainly, you know, some of our orthoptera or our orthoptera experts in natural England would certainly be using techniques like this. I also took part in the trial that Southampton University did to try and re-find the New Forest cicada, and that was using a phone app that used basically your phone would take a recording and the software would assess your recording to see if it had heard New Forest cicada. Sometimes it got very excited by jangling keys in your pocket, or sometimes planes that flew overhead would say, oh yes, that could be one. But then you sort of uploaded the recordings to the university and they analysed them properly. And unfortunately, they didn't find it. But I thought it was a really good bit of citizen science, so that anybody who went to the New Forest, walking the dog, doing whatever, you know, any sort of visit, going for a picnic, they could spend 5 minutes making some recordings and contribute to a sort of serious scientific project. So actually, that's the sort of thing that I might try and get into some of the science work that I do in natural England. So it would be great to get some of those similar sorts of projects underway.
Yeah, it sounds like a brilliant survey. I've not heard of that.
Yeah, it's probably a few years ago now, but yeah, it's really good.
Right. Okay. Another question from Ian Strahan. I do a lot of Malice trapping, but hardly anyone is looking at some of the most abundant groups, e.g. phorids, parasitica, smaller Nematodera.
Yeah.
I guess these aren't used in Pantheon, but must be important. What can be done? I'm trapping to get grips with some of these groups. Is a lot of material discarded?
That's a very good question, Ian. And I agree. A lot of what you catch in Malice traps is classed as bycatch, which is unfortunate. Phorids, certainly very few people work on phorids. Parasitica, as we know, are really, really difficult unless you're one of 2 or 3 people at the Natural History Museum. Yeah, smaller flies. These groups are certainly not on Pantheon at the moment. But then again, if you go back 5 years, there are, you know, quite a number of the groups that are on now weren't on there. So it will come in time. So things like fungus gnats, as an example, they're a really interesting group that tell us a lot about habitats, but they're not currently coded. and there aren't that many people who look at them. But that will come, and I think the process for that to happen is that we need to do IUCN status reviews of these species, and once there's a status review, then we know the which the rare species are, which the habitat specialists are, and then they can be coded into Pantheon. That will happen with fungus gnats sometime soon. The IUCN status review has been underway for a number of years, but it is getting closer. And as soon as that's done, then potentially they can be coded into Pantheon. Phorids is another matter. I think we're a long way off phorids being coded. In some parts of the world, though, for instance, in America, I was talking to a chap who works on phorids, and they've even had citizen science projects on phorids. So I think it depends where the experts are. If there are experts around who are able to, to work on those groups and get them coded onto Pantheon, then they become usable. So who knows? Watch this space. In terms of material being discarded, invariably some of it will be. I'm as guilty of that as the next person. I've used Malice traps a lot over the years, and there's just no way I could keep everything. Whether my local museum would have taken it at the time, I probably not. I would have thought just sort of random samples of stuff that nobody can identify. But in the future, when all these things are barcoded, then there may be a way forward. You may be able to get your Malice trap bottle, mash it up, not worry about what's in it, get it DNA barcoded, and it'll tell you the answers. It'll tell you everything that's in there. And that's probably where we're going within 20, 30 years, maybe not even that.
Yeah. It's with groups like phorids, is something holding it back from Pantheon? Is if we don't know the ecology very well, so the habitats of each of the species, does that stop it as a possibility for Pantheon?
It does generally. So if you look at a group of flies, for instance, say the muscids, which are a pretty big and complex group, but some of them are coded in. So those species where we know they're important, they're in there. But then there's a whole lot of other things, like say tachinid flies, that aren't coded in, even though we know them pretty well, because they're associated with other creatures. They're not viewed as includable, because their requirements are the same as other creatures that you might encounter. And also, finance is another issue. Pantheon has cost quite a lot of money over the years to get to the situation where it is now. It started off as a spreadsheet. Some of you may recall it. It was called ISIS. So it wasn't, that its name became rather unusable as events in the world occurred. But the ISIS spreadsheet is still the sort of basis of the current software. But micromoths weren't on ISIS, and they are on Pantheon now. But that took a lot of work to get the micromoths on there, because somebody had to assess them and code them and see which habitats they're associated with, do all the statistics, the analysis behind the coding. It's a lot of work and it's expensive. And we're constantly trying to get more money to develop it further. But it's a bit of a battle in the current climate.
Thanks, Pete. And Ian has just put another question in the chat there about any plans to extend Pantheon to Scotland. So is the finance there?
There are apparently talks that have been either going on or some form of contact has been made with the with Scotland and Wales to see whether it could be a wider bit of software that applies everywhere. One of Pantheon, I don't want to get into Pantheon too much today, but one of the problems with Pantheon is that it doesn't really recognise geography. So it was tested on a certain number of SSIs in certain parts of England where those habitats were richest. So for instance, heathlands, it does very well, it performs very well on Dorset heaths, but probably performs less well on heathlands up into Yorkshire. And likewise with fens, it does fantastically well in Cambridgeshire, but fens in Northumberland perform less well currently under Pantheon. But the idea eventually is to have it representing properly different parts of the currently country, hopefully countries, in future times. So it would be great if it covered Scotland, Ian. I hope it will do one day.
It's good to hear that there's, you know, that there are, you know, obviously plans to develop Pantheon further when possible. It's good to know that that the, you know, that could be invested in further. It's not going to, you know, it's a program that will last a long time. People can really get to learn and without fear of something else coming along and replacing it quickly. While we were talking about that, just to go back, for those not paying attention to the chat, Andy Musgrave just put in a nice link there. If anyone wants to analyse sound recordings of bush crickets, check out some of the work that his colleagues at BTO have been doing. So there's a link there in the chat.
Oh, fantastic. Okay.
So a question from Andy Marquez. I'm horrible at pronunciation. I'm really sorry. Thank you for the talk. Really good question. How could we deal with a lack of local expertise in areas which could be important to a site, e.g. Hymenoptera and arachnid experts are rather lacking locally? Not sure where you are, Andy, but
I think it probably applies to large parts of the country that aren't covered by either the Tanyptera project or previously work at FSC have done in the West Midlands. It's a matter of investment ultimately. The work we did at FSC on you know, similarly to the sort of stuff you're doing today, Gary, it cost us something like, you know, £250,000 to run that project over 4 years. So it's not cheap, and to get people up to a skill set that they can identify Aculeate Hymenoptera or spiders or any other group takes a lot of time. It needs a lot of one-to-one study with experts, as you know. It needs, you know, we ran our invertebrate challenge project over 4 years, and that was the level of time we had, and we had events every sort of, you know, 2 or 3 weeks through the year, and it needs a lot of time and a lot of input and a lot of resource, and either government or others in other parts of the country have to you know, get those funds to be able to do that. And also at the same time, bearing in mind it's not the sort of subject area everybody is into. As much as we would want to think they should be, we want lots of entomologists. We now, us entomologists think, why can't other people see this? Why can't they?
Exactly, yeah.
You know, why can't they do what we do, see what we see? But not everybody's into it, and there's so many other calls for finance in this day and age that it's difficult. But it does need an organisation to be able to, you know, to sort of either underwrite it or support it with funds and resources. But there are lots of precedents for it happening, as I say, through FSC, Tanyptera, and others. So it is doable. It just needs resource.
Yeah, thank you, Pete, and I hope you're wherever you are, Andy, that yeah, there is a future sort of similar project to what FSC are doing in your area. Now, BioLinks is, you know, their work they're doing at the moment with the FSC. They're covering quite a big area because they're also doing part of that project is southeast as well.
In
So
London.
yeah, so I don't know if you fit into that, but okay, thank you. So final question I've got here is from Chloe. How do you process specimens after they've been in pan traps in water? Do you put them into alcohol or just dry them out as soon as possible after trapping?
You can dry stuff out pretty effectively if it's been in a pan trap for not very long. So, you know, within a couple of hours, you can readily dry them out. And we've done that with aculeates, and they'll often revive as they dry out. If you, on the other hand, if if they've been in a pan trap that's had some preservative in there for weeks, it's a little bit harder, but you can still dry stuff out. I tend to, as I said earlier, I put most of my stuff in alcohol, and sort of, you know, as that starts to do the trick, and then you can change the alcohol after a while, because you invariably will have some water in it. And yeah, a number of alcohol changes will sort of do the trick. There are methods of drying things out. I tend to dry out most of my flies at the end of the year, so I will have my my pot, wherever it is, I photographed earlier. I'll have my pot with lots of flies in them, and I will tip them out, or I'll hoik them out and put them on a kitchen roll, and they'll dry out enough for me to be able to identify them. They won't be of the quality of specimens that way you would need to sort of pin and curate properly, but they'll do enough to identify, and so that's what I do. There are a number of other methods that you can use, but I certainly don't do that, and it's good enough for me.
Thank you, Pete. And I was going to mention, actually, earlier we were talking about Malice traps, samples, etc. That World Museum has got an awful lot. We've got a lot of stuff from over the years from Liverpool surveys that hasn't been thrown away, but it is in unknown piles, because even though there was especially going back 20 years, there was quite a number of entomologists working full time, you know, as curators of numbers of, you know, curators who've got smaller across the country, but even that wouldn't cover, you know, the whole range of stuff that they were getting in the Malice traps. So, and most of it is in IMS, so they don't think it's very good for DNA, but it may be solved, and it may be in the future be DNA-able. So, yeah, even though so any expertise of very obscure groups in this call right now, you're welcome to come and look at them. But I think we're reliant on perhaps DNA in the future to sort those out. So if there's no more questions forthcoming, you can speak one now if you like. Unmute yourself, or put a quick final one in the chat. Otherwise, you'll have to save it till next week. Next Thursday, same time, 7:30. And yeah, we'll get into Pantheon.